All right, I'm here to do a video on sensory function. This is going to be an introductory video covering basically general principles. Now, may even have more than one of these. Um, but what I'll do is sort of give you a perspective of what sensory function does, and then some of the categories of of stimuli that the receptors um, um, can detect, and then some of the anatomy of the different versions of the anatomy in that sensory afferent division of the PNS. And so sensory function is a little interesting because we have our own perspective of what our sensory function provides to us. And I think a lot of people, myself included, started out thinking, well, your sensory function is reflective of the real world. What you see is what's real, and everything that's real. And that's not entirely true, and not even remotely true in some cases. What that means is that evolution has provided sensory systems that are beneficial to survival. They're not adapted to reflect reality. And an easy example of that would be, our vision, where we detect electromagnetic radiation in the visual spectrum, right? There is enormous range of electromagnetic radiation outside of the visual spectrum that we do not detect. We don't see it or detect it in any other way. And so really what that means, and so for an example, there are some insects that see, wavelengths of light or electromagnetic radiation that is outside of what we see. So their version of vision, their version of of the visual world is different than ours. An even more extreme example would be to use bats, right? So bats have reasonably poor eyesight, at least some bats have reasonably poor eyesight, and hunt at night often. Again, some, not all. But the ones that do use a form of echolocation to quote unquote see what they're doing, to find their prey, to detect motion, to detect objects in their environment. And so their version of the world, vision, it's not vision per se, but it's a completely different sense, a mapping of the what we see in a way that's more like sonar than it is like vision. Is one better than the other? Not really. They're both functional for the adaptive purposes of the organism. So again, I don't want you to think that our senses are there to reflect reality. They're there to essentially keep you alive, right? They're there to detect stimuli in the environment. They'll allow us to find food, to avoid threats, things like that. And we even saw some of that with reflexive actions that were generated that were protective before, right? Okay, so we don't want to think, right? So we've already got our sensory function in the category of detecting information that's beneficial, but not necessarily complete, right? So let's go through some general principles of sensory information, and we already know that requires receptors. And oftentimes, but not always, those receptors are neurons themselves. And so we need receptors. The way they do that they detect stimuli, and that can be environmental stimuli or it could be internal stimuli. And again, we'll categorize the divisions of the sensory system. By an external stimulus, I mean things like light or sound or touch, right, from another person or from an insect landing on your arm, anything like that. Internal stimuli would be things like, what is your blood pressure? Is your stomach full? Is your bladder full? Those would be internal stimuli. What's your blood oxygen levels? Those are also detected. And they detect all of these potential stimuli that come in a variety of forms that I'll lay out in a minute. But what they do is they take that stimuli and they convert it, which is called transduction. Right? So they're transducing it, converting it or transducing it. That process is called transduction of a stimuli to a change in VM. Remember, the delta sign means a change in VM. VM being the voltage of the membrane, right? So changing a membrane potential. So what that means is that anything, whether it be light input to photoreceptors in your eyes or tactile touch on your skin, has to be converted into electrical signals. That is just how our nervous system both receives and then transmits in a different form, in the form of an action potential or a series of action potentials, a nerve impulse, to the central nervous system for processing. Guess what? The processing also revolves around connections between neurons that are having changes in membrane potential, right? And so, how do we get this change in membrane potential? How are we transducing it? And by the way, that change in membrane potential is called the receptor potential. Right? In other words, if a stimulus arrives, is transduced into an electrical signal, that electrical signal that it's being transduced into is called the receptor potential. And I'll point that out occurring when I draw a sensory neuron here in a second, sensory neuron. And so, but before we do that, let's talk about the types of receptors we have to detect these stimuli, because you think, wow, there must be an enormous number of them, right? Because there's so many different types of stimuli that you could receive. Well, that's true, but they fall into essentially 4 main categories, and 5 if we sort of use one that overlaps a little bit. So these would be types of receptors, or receptor types. Right? And again, all receptors fall into one of these categories. Well, an easy one is a photoreceptor, right? Detects electromagnetic radiation in the visible spectrum, right? The rainbow of colors, essentially. There are mechanoreceptors, right? These are the receptors that detect mechanical distortion. They detect pulling and pressure and vibration and stretch and any of those sort of like physical distortion terms that we might throw onto that. Right? So things like tactile skin receptors will be many of them, not all of them, many of them will be mechanoreceptors. Stretch of the walls of your heart or blood vessels or bladder or GI tract, right? All of those would be mechanoreceptors. There are thermoreceptors, right? Detecting the obvious changes in temperature. And remember, they're always generating a receptor potential, but for us to perceive these stimuli, they have those graded potentials. The change in VM, the receptor potential is a graded potential, has to then be initiate the generation of action potentials, right? Because if you don't get action potentials, you get no transmission. Right? If a small enough insect lands on your arm, you don't notice it because it doesn't reach the threshold of detection. It won't generate action potentials. Right? And so all of these receptor types are taking these stimuli, converting into receptor potentials, but that doesn't necessarily mean that you're going to detect it. Right? Because again, you have to breach that threshold to open the voltage-gated channels to get action potentials. All right, so we got photoreceptors, mechanoreceptors, thermoreceptors. What else do we have? Chemoreceptors sense the chemical environment. The easy examples are blood oxygen levels or blood carbon dioxide levels or blood pH levels. Those are all chemical sensor, chemoreceptive um, sensors. There are others like the sensory receptors in your sense of smell or on your tongue for taste, right? Those would be chemical receptors as well. We have some in the walls of our GI tract to sense the presence of food, particular nutrients, right? So there's a variety of them found in particular locations. And the last group, so this is the four big groups. The last group is called a nociceptor. Right? And again, the hyphen, photoreceptor, mechanoreceptor, thermoreceptor, right? So nociceptor detects pain, right? Now, pain it's not really its own category. It's not really like the first four, because there are nociceptors that detect thermal extremes. There are nociceptors that detect chemical changes. The nociceptors in the mechano, right? Distortion. That's an easy one, right? Somebody punches you in the arm. Those are mechanoreceptors, but if it's painful, you're also stimulating nociceptors that are detecting that mechanical, distortion. The only place there wouldn't be a subset here would be photo. Now, these receptors are going to detect, and again, if you reach the threshold, convey information of a single type, and that type of info carried is called its modality. Right? So each type of receptor receives information in a given modality. Now, how we funnel a particular stimulus into one of these receptor types is dependent upon a couple things. One of them would be the location. In other words, your olfactory receptors are in a very specific location in your olfactory epithelium in your sinuses. And so air we breathe in is what we're sort of sampling with those receptors. They're not on the surface of your skin, so they're not sampling the air out here, right? You're running the air, across them more dramatically and regularly by putting them in a particular location. Same thing with photoreceptors, right? They're in a particular location. We can also funnel information by the type of channels that get gated here. In other words, what opens a mechanically gated channel does not open a chemically gated channel, and vice versa, right? So the channels are very specific for the type of information, the type of stimulus that's arriving. And lastly, there are often filters. These filters, we'll talk a little bit about that later, but the filters can sort of hone the type of information or the type of stimulus to that particular type of receptor. The special senses often have filters that help sort of funnel information and filter information to those receptors. And again, we'll talk more about those later. So each type carries a single modality, and they carry that modality along what's called a labeled line. Right? Lines. So labeled lines, so this is sort of like saying, your photoreceptors in your eye, right, are only receiving, and they have filters, lenses and things like that help funnel that light information to those photoreceptors at the back of your eye. So they're going to receive that single modality, right, the photo information, visual light. And then they're going to carry that information along neural pathways that are called labeled lines. So in other words, and actually, to complete that, they're going to carry that single modality along a labeled line to a processing site in the cerebral cortex that can only process that type of information. In other words, your visual cortex can only process visual information. So just as a good example, easy example, right? You wake up early in the morning and you rub your eyes. And if you rub your eyes too hard, even in the dark, right? It's a dark room, you got your hands over your eyes, and if you sort of rub your eyes too hard, you'll see flashes of light, right? So what you're essentially doing there is you're generating pressure on your eyeballs that is stimulating those photoreceptors, right? Abnormally stimulating them. But the only way that those photoreceptors can sort of interpret that information, that excess pressure, is by generating a signal that's read as that fixed modality, and they're going to carry it along the labeled lines to your visual cortex, and you're going to see flashes of light. You're not going to feel, you may feel pressure because from other receptors surrounding the eyeballs, but the eyes themselves are only going to be able to convey a single modality, visual reception, along those labeled lines and process it as light, right? That's the basis of these specificity of the receptors sort of detecting one thing, carrying that type of information, and then processing only that type of information at the visual cortex. Okay. All right, general things there. Let's see. So let's talk a little bit about the cells. We've already seen them before. They're called unipolar. Actually, to be more specific, they're called pseudo-unipolar. So the sensory neurons look a bit like this. I mean, pseudo means fake. And the idea there is that the axon is set off to the side of the soma, but then it branches, right? And so it's actually going two directions from that one pole off of the soma. So anyway, it looks a bit like this. Okay. Right. So there's our pseudo-unipolar neuron. And so that's the soma. Right. This is axon. And this is axon. Right. So the axon kind of runs past the soma offset to the side there. And there's actually a peripheral axon, this part, and a central axon. Not all that important, but if you think about it, it makes sense because, and I'm just going to draw a sort of a red line here to indicate a break, right? Because as we've seen before, the cell body sits outside the central nervous system, right? In a sensory ganglion, right? The dorsal, on the spinal cord, the dorsal root ganglion, right? There are ganglion on cranial nerves as well. But so this is CNS, and this is PNS, right? And that cell body would be found in a peripheral ganglion. And so this is the afferent, right? Information's going that way. Afferent incoming information, sensory information from out here, again, in periphery. Well, periphery, right? Could be external to your body for external stimuli, but it could be internal as well. Think about lining of your GI tract and respiratory tract and bladder and any other, blood components, all those kinds of things, thermoreceptors in your torso or in your skin, right? And these are essentially the dendrites, right? But we don't call them dendrites because they're not on the soma anymore. But they really are, but we're going to call them the nerve endings. Right? And so these nerve endings are going to have receptors that detect one of these types, these modalities of sensory input, right? So they might have a chemical receptor for oxygen or carbon dioxide or a particular nutrient. They could be mechanical receptors, meaning they would have mechanically gated channels that are opened by mechanical distortion, pushing on this, right? And so again, depending upon what this input is, determines what type of receptor mechanism we need here to generate the receptor potential, right? So we're going to get a change in VM here. Here comes our stimulus. right? Based upon what type of sensory neuron this is, right? And these categories, it will have a mechanism of detecting this stimulus or not. If it's a different type of stimulus, it doesn't detect it, right? Remember I said mechanoreceptors do not detect chemicals, right? You ignore it, right? There's no mechanism detection. But if they match, right, then our stimulus is going to generate a change in Vm, delta Vm, which is our receptor potential. And then right about here, right, sorry if you can't see that, but that's that little block of red I just made is, you should know this, the axon initial segment, right? That is where we now run into voltage-gated channels, right? And so our graded potentials traveling by electrotonic current, passive, right, decaying with distance, along the dendrites here, along the nerve endings, hit the axon initial segment. If we don't reach threshold, nothing. You have no perception of whatever this stimulus was. If you do reach threshold, you now generate action potentials. They'll travel all the way to the end here, right, into the CNS, to the synapses here, and then we could add interneurons sort of going off and synapsing on whatever they're synapsing on, right? Going up to the brain, generating motor output to respond, whatever and all of the above, sort of completing the pathway. Okay, so we've got our pseudo-unipolar neuron. Now this is one version of a sensory neuron. The other, so this would be called, these in this particular picture, these would be called free nerve endings. Those are unencapsulated nerve endings. Remember I said before that sometimes there's a filter that helps sort of like minimize and focus what these nerve endings would receive. So sometimes they have this connective tissue, usually it's connective tissue, but a connective tissue sort of ball around it that helps to sort of like, it's like a cushion essentially, and it'll determine what stimulus will affect those nerve endings, right? So encapsulated nerve endings are often mechanoreceptors, like tactile. So there's free nerve endings, there's encapsulated, unencapsulated, there's encapsulated nerve endings with have a specific filter on it, connective tissue filter. And then there's another version, which is both of those versions, free nerve endings are encapsulated as a single neuron, sensory neuron system. In the special senses, it's a two-cell system. So in that system, you have a cell, let me make it a different color. And I'm going to give it some microvilli looking structures here. There's a nucleus, right? That is a receptor cell. Sometimes it's a neuron or a modified neuron, but sometimes it's a modified epithelial cell. It's not even a neuron. But in this system, this guy is going to release neurotransmitter upon having a stimulus, receiving a stimulus. Right, remember this is special senses. So all special senses are two-cell system. So the receptor cell right, will release neurotransmitter when, again, after a receptor potential. It'll release neurotransmitter, right, in response to that graded potential, onto our pseudo-unipolar neuron, which looks much like we had it look before. Right. And so now our axon initial segment is here, right? That's our sort of modified receptor dendrite region there. Again, because they're not doing the receiving of the information, this specialized cell is. So all this has to do now is receive and bind the neurotransmitter here. Again, if you don't release enough neurotransmitter here to reach the threshold of the voltage-gated channels, no detection, right? If you do, then all of a sudden we've got action potentials here, right? Axon initial segment again, and all of a sudden we're sending our information into the CNS to be perceived, to generate perception. Okay, so general senses of the somatic and visceral categories would be, of the first two, right? The either free nerve endings or encapsulated nerve endings. Special senses always are a two-cell system. I was gonna say something else there. Okay. One other thing I wanted to point out were those categories, which is I'm running out of room down here, so I'm not going to write them out. But the categories are, and we saw this on day one, right? When we categorized the nervous system in general, we said CNS, PNS. PNS is divvied up into the afferent coming in, sensory, efferent coming out, motor, and then the sensory, to be specific for that section, the sensory is divvied up into special senses, somatic senses, and visceral senses. We'll spend most of our time talking about the special senses, just because we're more familiar with those and there's more intricacies to those. But I'll do a video on the general senses as well. Somatic senses are our sensory perception. Remember, somatic means referring to your body, and even more specifically to your skin, bones, and muscles, really, right? And so we want to, in the somatic sensory category, we want to put tactile skin receptors, temperature, pressure, pain, right? We want to put joint position, right? That's called proprioception. And we want to put, and also pain and things like that as well. And so, right, so somatic senses help us detect where our limbs are in space without having to visually process where they are, right? So you can close your eyes and touch your nose and it still works, right? Because I know where my limbs are in space based upon joint receptors, muscle receptors, tendon receptors, skin receptors even. They're telling me, how my limb is bent even, right? That's all somatic sensory. Visceral sensory is sensing the viscera, which is sort of a term for organ function or organ sense. So visceral sense would be things like distension of your GI tract, distension of your bladder. Again, pain, right? You can get visceral pain, right? You had somatic pain, but then you remember pain was kind of a crossover, right? So you have somatic pain in your skin or your joints, but you have visceral pain in your, with your organs, stomach ache, something like that. Thermoreceptors, chemoreceptors, sensing blood content, right? That would all be visceral sense. And then special senses are the ones that are associated specifically with the head: vision, hearing, smell, taste, and equilibrium. Equilibrium being of inner ear function. Okay, I think we've got things well categorized. I'll probably do another one that talks a little bit more about the possibilities of wiring, how the information is processed. We'll go from there.